This is a production of Cornell University.
Thank you. Yes, as Emily said, my name is Erin. I'm a fourth year PhD student in my course lab, and I'm excited to talk with you all today about integrating data collected from different proximal remote or proximal sensors for improved prediction of important traits. So proximal sensors present a mechanism of field-based high-throughput phenotyping that saves time and labor as compared with manually measuring phenotypes. So proximal sensors are those which do not come into direct contact with the plant, but are more close by or proximal than remote sensors such as satellites. So you can see here some commonly used platforms for field-based proximal sensing. These include unoccupied aerial vehicles such as drones, gantry systems, tractors, push carts, ground rovers, and even smartphones. The sensors which can be deployed on these different platforms include typical red, green, blue cameras, multispectral and hyperspectral cameras, which include additional bands beyond those. And then we also have LiDAR, thermal, and radar, which can sense below ground traits. So these sensors have a broad application to both breeding and precision agriculture. They can be used to forecast end-of-season traits, therefore allowing selections to be made pre-flowering and decreasing the breeding cycle. Features from these data streams can be used in phenomic prediction, where those features replace traditional markers in genomic prediction. They can also be used for building populations for genomic prediction, genome-wide association studies, and the increased temporal resolution lends to studying genotype by environment interactions. They've also been used for stress detection and efficient allocation of resources in precision agriculture applications. So due to the kind of broad-reaching applications for breeding programs, significant investment in studying the use of these different sensors and platforms for a variety of different traits have been carried out in recent years, including productivity traits such as yield, biomass, quality traits such as taste, and many others across a wide array of crops. What this has ultimately led to is a large number of crop and trait-specific algorithms. In addition, these data streams are both very high-dimensional and complex. A single multispectral image can have a couple 100,000 pixel values for a single plot. And so how to effectively extract meaningful information from the data becomes the challenge, and we have ultimately relied on manual curation of features. In addition, you can see there's a variety of different sensors which capture wavelengths across the electromagnetic spectrum, and platforms which allow capture of information at canopy, subcanopy, and below ground scales. And while there's likely unique information contained within each of these data streams, this data is usually used separately from each other, and integration is uncommon and lacks generalizability. So as an example, on the left here we have a kind of pictographic representation of a multispectral image, which can be thought of as a grid of pixel values for each band, the bands kind of stacked on top of each other. The most common mechanism for extracting information from these is by calculating vegetation indices. So you might be familiar with the normalized difference vegetation index, which is calculated by taking the relative difference between the near infrared and the red bands of the image. Ultimately reducing down to a single value per multispectral image. An alternative to this is latent phenotyping. So latent phenotyping is a mechanism of extracting features or information from proximal sensing data streams using unsupervised machine learning algorithms such as autoencoders. So while NDVI in this example takes in just the NIR and the red band, we're able to pass the entire image into the model to extract information. In addition, where NDVI reduces down to a single value, we can extract a larger number of latent features. We specifically do this using autoencoders, which can be conceptualized similar to principal component analysis without the linearity constraints. So it takes the input image and passes it through an encoder to reduce the dimensionality and find an embedding. That reduced space is then passed through an analogous network, the decoder, to reproduce the original image. And the model is optimized such that the difference between the original and the reconstruction is minimized. So in this way, the latent features should contain the necessary and sufficient information to reconstruct the original image and are hopefully the most relevant then and important features, parsing noise and other unimportant information. So our goal is to make use of latent phenotyping to perform integration across different sensors, preserving additional information that is lost with traditional mechanisms of feature extraction, and providing a generalizable method of analyzing these data. So I'll go through our end-to-end pipeline of data collection, processing, and analysis through extracting and integrating latent features. So we have specifically focused on multispectral images collected by unoccupied aerial vehicles or drones and LiDAR scans collected by unoccupied ground vehicles or the rover here. So the multispectral images collected blue, green, red edge, and near infrared bands, capturing that canopy level spectral information, which is commonly extracted as vegetation indices. The LiDAR scans can be used to generate three-dimensional reconstructions of an entire plot, giving sub-canopy architectural information. These data were collected for maize hybrids planted as part of the Genomes to Fields initiative for weekly collections from 2020 through 2024. For each growing season, we pause rover collections a couple weeks post-flowering until senescence as structural changes slow during that time. Ultimately, giving a data set of about 75,000 plot level multispectral images and about 35,000 plot level point clouds. So the multispectral images are collected by this drone here. You can see the camera on the bottom. The raw images have about 80% overlap with each other, which allows them to be stitched together to generate a field scale image or orthomosaic. The field scale image can then be split down into our individual plot level images. The LiDAR camera is on the back of the rover here, which drives in between the two rows of maize. The LiDAR scans collect the distance emitted light has traveled to the nearest object. It can be thought of as being in polar coordinates. This is essentially converted into Cartesian coordinates to generate a three-dimensional point cloud where each point has an X, Y, and Z coordinate associated with it. Do further processing on this to remove erroneous points. Do vertical flipping, it's kind of stacking the rows on top of each other and then compressing that down to a single two-dimensional image or density map, which can be input into an autoencoder. So we have trained autoencoders for both the multispectral images and the 2D density maps. You can see examples here of original images and as well as their reconstruction for both the MSIs and density maps. The latent spaces can then be extracted from each of these trained models and integrated. So once these models had been trained and latent phenotypes had been extracted, we wanted to first see what kind of information was contained within them. So these are latent phenotypes for a single field in a single year. We calculated the heritability for each of them in LiDAR and MSIs. And what you can see here is the heritabilities range from near 0 up to about 0.9. So we're capturing both heritable genetic information as well as is likely environmental information. The latent phenotypes, once extracted and integrated, are then used for characterizing manually measured phenotypes for the G2F fields. So for each of the 8 ground truth phenotypes that were collected as part of the Genomes to Fields project, these were predicted by MSI latent phenotypes for all dates in the growing season, LiDAR latent phenotypes for all dates in the growing season, and then the integration of the two. And these were compared against a baseline of 49 different vegetation indices, extracted or calculated for all MSIs throughout the growing season. And what you can see here is that flowering time and grain moisture have higher, NDVI has higher predictive ability for those traits. For the remainder, the integrated latent phenotypes have higher predictability. With variation on when MSI or LiDAR latent phenotypes are more highly predictive. For example, you can see here ear height and plant height have higher predictability from the LiDAR latent space as their architectural traits, if it makes sense. Overall, we see a 4.6% average increase in prediction accuracy for the integrated latent space over vegetation indices across all traits. Similarly, the integrated latent space outperforms MSI and LiDAR latent phenotypes by an average of 5.1% and 20.8% respectively. So finally, we wanted to look at how the prediction accuracies changed with the addition of more information throughout the growing season. So we iteratively added dates of information for each of the 8 phenotypes here. So as predicted with the integrated latent space, we can see some prediction accuracies don't change. For example, stand count, which isn't changing over the growing season, so makes sense. Some traits which are not very easily predicted early in the season and only see kind of a large bump toward the end. But notably, there are some indices and traits that have reasonable prediction accuracies pre-flowering. So yield here sees a 0.1 increase from the pre-flowering predictions to the end of season, incorporating all of the dates, indicating the potential for using this data for pre-flowering forecasting. So I would like to thank my committee, Mike Bohr, Aliyah Tufan, Ying Sun, and Margaret Smith. I'd like to thank the Gore Lab and Buckler Lab, specifically Liam Wicks-Doe and Nick Pack for helping with the efforts of collecting so much of this data over 5 years. Kelly Robbins and Joe Gage for their assistance with modeling. Abe Davis, Peter Michael, and Zeshi Gu for their assistance and incredible work on the autoencoder modeling. So thank you so much, and I'll take questions. Yes, Charlie.
A lot of the traits you're looking at these correlations with are the traits themselves are correlated with each other, right? So like, flowering time and yield. So is there any way to like kind of disentangle which of those the models or which of those the latent spaces kind of capturing and which are like second order correlations?
Yeah, that's a great question. So the question was, since so many of these phenotypes are correlated with each other, is there a way to disentangle which the latent phenotypes are capturing and which are just correlated with each other? Is that the question? So I think that is a great question, and we haven't done a lot of investigation into which latent phenotypes are more highly correlated with which traits and what they're capturing. Ultimately, the latent phenotypes are still kind of a black box for us, and maybe relying on these relationships between traits. It's kind of a secondary kind of selection, but that is a very good question. I'll definitely look into that more. Tim.
When we fly drones, we have to near solar noon, and you have to have consistent cloud cover. For the LiDAR rover, are there similar constraints, and do those impact how well certain data points can be integrated into your model?
So the question is, for flying drones and collecting multispectral images, there are constraints on time of day and cloud cover and other conditions. Are there similar constraints for the rover data? So insofar as the data collection quality, there aren't really constraints. The constraint would be around when we can feasibly drive the rover. So if it's rainy, of course we can't, or if it has rained heavily in the past day or two and it's too muddy, the tires will, just kind of spin. So more constraints on the platform itself than the sensor. Yeah. And
yeah, very nice. What I was wondering about, have you tried training between the two reps? It'll mess up your heritability estimate, but if we want to say we wanted to capture really about genetics and kind of ignore some of the environmental ones, have you been able to? Does that work, or have you tried that yet?
Yeah, so that's a great question. So the question is training between reps. So using one rep as the input image and the second rep as the output. Is that correct? Yeah. So we haven't tried that. So we've been focusing on kind of plot level predictions themselves, and have looked less into kind of parsing out those genetic and environmental factors within the latent features. But that would definitely be something that we should look at for the next iteration.
It would also be interesting to see if you could predict LiDAR, use the LiDAR to predict the other one, the MRI, and as essentially make the embedding space in between those two or something like
that. Oh yeah, that's a good point. Thank you.
Yes. I didn't quite file for latent phenotypes. Is there an intuitive way you describe those in terms of okay, they're kind of analogous to this in a PCA, or like this in weights in a layer, or so they're dimensionless metrics on a plot level versus or an image level?
Yes. So you can think of them as the principal components of some higher order object. That's probably the closest analogous, kind of conceptual way of thinking about it. Yeah.
I'll do the last question here. So I know that, of course, the labs you work with maze, but I was wondering with the rover, are there any kind of architectural limitations about what crops that you can drag them through?
Yes, that's a great question on the limitations of the rover depending on the architecture of the crop. So the resolution of the LiDAR scans is on the order of centimeters. So anything that is too small would not be able to be resolved by at least the quality of the LiDAR camera that we are using. There are likely higher resolution ones that could be used. Also anything where you have what you're trying to measure may be blocked by kind of lower foliage or something that might, because the LiDAR is going to bounce off of whatever it hits first. So if you have something where you have lots of leaves or something and you want to maybe get imaging of fruit that's up higher, the rover might not be the best application. Yeah. Thank you so much.
Thank you very much. Good afternoon, everybody. My name is Siim, and I'm a third-year PhD student at the Small Grains Lab with Professor Sorrells. And today I'm going to talk about organic naked barley and breeding innovations for resilience in the state of New York. First of all, about naked barley itself. As one could imagine, it's the part of naked in there means just the loss of the barley hull itself during the harvest process, which is something that occurs naturally thanks to the loss of function mutation in the new gene 8,000 years ago. And this gene apparently coats the lipid layer that would glue together the hull and the caryopsis. And it's visible here, as we see in the quick laser out here. On the left, we see the covered barley, and on the right, we see the naked barley or the hull-less barley. And there are two big benefits about that. So one of them is that when we have covered barley, we are relatively limited with the use of that, as if we would like to use covered barley in the food products, we would need to go through lots of processing steps to remove the hull as it's digestible. And with that, we could also lose important and nutritious parts like the bran or the germ of the seed. But with the naked barley, this is all going to stay in the grain after our harvest, and we're not going to lose any of those micronutrients. And this allows those multi-purpose uses for the naked barley and making it more digestible. And all of this together kind of indicates our potential for organic and small farmers as a healthy and an easy to process alternative in their cropping system. And this is where we come in with the multi-use naked barley project for organic systems. That's a collaboration between 5 universities, Oregon State, UC Davis, University of Minnesota, University of Wisconsin-Madison, and us here at Cornell. And we have quite a wide variety of goals we're trying to achieve with that project. Most of all about educating the public about naked barley itself and the health benefits of that, combined with working together with stakeholders to build those markets for these naked barley products out there. While at those different universities, we test the existing germplasm and also generate new genetic diversity in the face of, or like as an example with our breeding lines that we're trying to get out there that would be locally adapted to all of those environments that we would like to see the naked barley be grown. So for this, we have different kinds of populations in our hands to be used. As in, we conduct regional trials across all of those locations. We have a diversity panel to look into the genetics in a much more fine level. And we also do have the breeding populations that we are growing across all of those locations to be able to select for local adaptation. And talking about local adaptation, the most important trait we're trying to implement here in our breeding process in upstate New York is pre-harvest sprouting resistance, which I'm gonna tell more on the next slide a little bit. So pre-harvest sprouting is something that can be quite devastating for barley farmers. As an example, as a definition, pre-harvest sprouting is something that's defined as a germination, unwanted germination of the barley grains in the spike right before the harvest. and as a result, causing damages to the grain quality and the starch and the enzyme profile, which could end up with a complete yield loss and losses of profits for the farmers. And from the perspective of a breeder, it is a relatively expensive and complicated trait to screen. As we see here, this is the scale. We do screen this trait in our breeding program. But for this to happen, we need to sample the spikes, dry them out, and then mist them 3 days in an artificial misting array in our greenhouse to be able to score the level of germinated grains in a spike from 0 to 9. But luckily for us, we already know that a lot about the genetics of pre-harvest sprouting from the work previously done in Sorel's lab by several grad students. And I'm just going to bring some of those examples out here. First of all, we had a former grad student, Dan Sweeney, being able to map the genetic control of pre-harvest sprouting in covered background, going from the Q-tail to the gene and also out to allele levels. As we see here as an example, that barley lines having 2 dormant alleles in this Q-tail have an average pre-harvest sprouting score of 3, but the barley lines having 2 non-dormant lines have an average sprouting score of 7. And some work from another student, Travis Rooney, who was able to prove the large effect Q-tail having an heterogenic effect in the whole background, and also how using those large effect Q-tail alleles as fixed effects in the prediction models improve the accuracy. So this has motivated us to tap into the genetic diversity of the same genes and alleles in the naked germplasm. And here is where I could come in with my proposed ideas for an experiment with the naked barley breeding population, growing those same lines out on the field in 2 locations, having them genotyped with a USDA SNP chip, and phenotyping all of those lines in a greenhouse to measure their pre-harvest sprouting resistance. And that would allow us to tap into the genetic diversity we have in the naked barley germplasm. That would also enable us to verify what we already know in the covered germplasm, like the genes, the allelic effects, and how often do we find those alleles in these gene pools. And also just to test out the predictive abilities with the naked germplasm. And as an outcome of that, we do gain a nice insight through verification in the naked barley background, and also broadening the overall understanding of what's happening in there, and allowing the breeders to use them in the future for their selections. In this, as an example, for all those different breeders in the same program, and being able to advance the materials in our program much faster with a good predictive ability from the model, and also being able to combine those local adaptation traits together with those beneficial traits already in the naked barley to provide good varieties for local producers here in upstate New York to grow them out. And here I would just like to thank all of our collaborators working with this project. And yeah, if any questions, I'd be happy to take them. Thank you.
So I was wondering, for your populations with the additional germplasm and SNP chip, are you hoping to find maybe more genes associated with the resistance to pre-harvest sprouting, or more so new alleles that might completely deter?
New genes, it's not that likely to. So the question was if I'm hoping to find new genes or new alleles in this naked barley gene pool that might not be present in the covered barley gene pool. And my answer to that would be, I think it's relatively unlikely to find new genes, but the diversity of alleles already present in those covered barley Q-tails it's pretty wide. I think for both of those Q-tails together, we already have 6 different alleles segregating in those. So some alleles and allelic variation is highly likely to be there that I'm looking to see. Yes. Another question?
Is pre-harvest sprouting correlated or related to any other traits you might be interested in? Cooking time, mycotoxin, post-harvest accumulation, macronutrient profile or anything?
It is So the question was if pre-harvest sprouting susceptibility or resistance is related to other quality traits. And as far as I know, we haven't tested them here at the Cornell, but we do also have collaborators in Oregon State who have a baking quality lab. And they have found some signs of deteriorated baking quality as an example of like early on pre-harvest sprouting that has deteriorated the quality of the bread or the dough they make out of it. So yes, I think it's a pretty clear relationship with that. Any other correlations or like what I've seen in the field? What I've seen in the field is relatively hard to see, like in terms of like, you know, what you see in the canopy or the spikes themselves.
Question on Zoom. Recats, is naked barley more susceptible to insect damage?
Not that I know of, and we have not seen that. So naked barley is more susceptible only to the smut disease because the hull is, I guess, like more loose, and I guess some of those particles of this fungi can access the grain in those early stages much better. But with other diseases, we haven't seen any difference with covered barley or also, yeah, with insects.
I guess, is there a categorical difference in terms of nutrient profiles, like how hull-less oats are 3-4 percentage points higher in protein, or anything like categorically different between naked and hull-on barley?
Yeah, so the question was about categorical differences in the profiles of naked barley and covered barley. And one of those big differences is the beta-glucan, which is actually something that is, the FDA has suggested for people to consume more as it improves the digestion of people. So this is one of those big differences, but at the same time, it's good to keep in mind why it's low in the covered barley, because beta-glucan can inhibit some of those malting processes and in the making of malt. So it's, even though we want to develop those multi-use purpose varieties, this is kind of a hard path to choose from. Like, I think with this, we either need to develop separate varieties, one for malting and one for like, you know, really high beta-glucan. That would, we could advertise them like this, yeah.
Yeah, my name's Aliyah. I'm a second-year PhD student. All of my research has to do with downy mildew resistance in grapevine. Yeah, so with over 45 different haplotypes that can confer downy mildew resistance in grapevine, there is a lot of stuff to phenotype. How do I phenotype it all? Is there a laser on here? Yes, with the Blackbird robot. So basically how the scheme works is we go out, we collect leaf from germplasm, either from the field or from the greenhouse. We use a cork borer, hole punch them out to create these arrays of leaf discs. That's also pictured here. These leaf disc arrays will get inoculated and then put into the Blackbird and imaged over a number of days to capture the disease progression. These images are uploaded to a convolutional neural network that will pick apart the images and give us a nice quantitative data set about the disease severity on the leaf discs. I can't talk about the Blackbird without showing you the beautiful images that come off of it. So this is grapevine downy mildew, which by default has to be my favorite pathogen. So my whole first chapter is a metadata study on the Blackbird robots. We really wanted to investigate the trade-off between this high data acquisition with the data quality to make sure that we're conserving our data quality. And we wanted to know if the system was really robust to the types of human error or data variation that could happen in the scheme. So basically what I had done was I have 3 real data sets that have come off of the Blackbird that have resulted in disease resistance QTL publications. I've simulated a bunch of variation onto those data sets, and I basically observed how the significance of the LOD score changed with those added errors and variation. And I did things like leaf disc swaps, leaf disc frame shifts to kind of capture the types of human errors that could happen in the arraying process, a different variation in disease severity to capture the types of microenvironments that are happening in the trays that might impact pathogen growth, and also replicate subsetting, because usually when we do these experiments, we have up to 8 to 10 replicates per experiment. So what did I find? This is an example of an intermediate effect size QTL. And all of these colored boxes represent the different flavors of variation that I introduced in the previous slide. Blue has to do with variation in pathogen growth. The pink is the leaf disc swaps, the orange is the leaf disc frame shifts, and then the yellow is the replicate subsetting. So basically you look at this and you say, wow, the Blackbird is very robust to the types of human errors that can happen, but it seems to fall apart when we pick apart the replicate number. That's great. Moral of the story, we were able to figure out that we are not compromising any data quality by our large volumes of data acquisition. This is supposed to be a lightning talk, so I understand that this is a great overview of my chapter, but happy to report it's been accepted to a focus issue of phytopathology. Literally this morning, updated the first look copy, so I'll make sure it gets in the newsletter. Very excited and happy about this. So yeah, what's the catch? We've all been slighted by technology, okay? There's veteran academics probably on the Zoom rolling their eyes at me, like I'm praising this robot and for what? There's gotta be something. And I'd like to introduce the bane of my dissertation, leaf hairs. So the Blackbird is based off of RGB imaging, so all of the visual wavelengths that we can see. So if there is morphology that is obscuring the phenotype that you're trying to measure, in our case, leaf hairs covering up the sporangia of the downy mildew, the robot can't see it. So how I plan to circumvent our leaf hair issue is with a hyperspectral Blackbird. This robot is called the Hyperbird. It's a lot less pretty and a lot less user-friendly than the Blackbird because it's a prototype. But I'm hoping to use hyperspectral imaging to kind of peer through the leaf hairs. LOL, I wish it was that easy. We saw from Erin's talk that hyperspectral imaging is really complicated. So what does this actually mean? Well, I'm hoping that the biochemical makeup, oh my God, of the host versus the pathogen is like distinct enough to where they have different hyperspectral signatures that will allow me to differentiate them on a microscopic scale. And it's a non-destructive method, which is really helpful to me, especially when you're phenotyping things like disease resistance. Yeah, my hopes and dreams for the Hyperbird. Number one, we have to ensure that the Hyperbird works. Again, this robot is a prototype, so we've never actually published anything with it. And I hope to do that with the actual leaf hair themselves, so hopefully they'll have a use. And we have a convolutional neural network on the Blackbird that can also measure leaf hair, so I'll probably compare it to that. Probably leaf hair will be conserved across Vitis, considering a lot of different Vitis species have the same leaf hair morphology. And then after we prove that the Hyperbird can work in this phenotyping scheme, then I can do the thing that I actually want to do, which is discover new disease resistance QTL on hairy vines. And then maybe I'll even take it a step further for this last bullet point, make some inferences in the relationship between leaf hair and resistance. This is supposed to be a lightning talk. These 3 question marks symbolize the 10-minute rant I could go on about this concept of leaf hair and resistance, and does it confer resistance and does it not, and is it a barrier and blah, 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 blah. Anyway, I'll save you the rambling. The important things are there is conflicting literature, and I'm very skeptical of some of the phenotyping methods that these papers have. And we might be able to solve some Labrusca mysteries as well. All of these hairy leaf discs that I've shown you, they're Labrusca. Concord grapes are a Labrusca variety. And basically these grapes have been resistant to downy mildew for over 200 years and no one knows why. Is it because of the leaf hairs? Is it because of an unknown QTL? We don't know. Maybe I'll find out. And with that, I would like to acknowledge my whole committee and their labs, especially Katie Gold and her lab and all the engineers that contribute to all these robotics. I'm housed at the Agritech campus in Geneva. Sirius Subcoda, he's been really integrated into all of my dissertation projects. And to Vitis Gen 3, not only for funding me, but for also just being an amazing research group that is very collaborative and supportive of each other. And with that, I will take any questions you might have. Yes?
So you talked about one of your greatest frustrations that you've been experiencing with the leaf hairs. I was just wondering if there's a great joy or something exciting that you had in the research.
Okay, I've complained a lot, and now Sam wants to know something that makes me excited. Okay, leaf hairs aren't all bad, okay? They, you can kind of see in my one picture here, they have a great diversity. They make the grapes really fluffy and cute. I don't know, I really like all of the research that I do. And me and Lance had a conversation a while ago, and he was like, oh, what's your favorite chapter? What's your favorite project? And I feel like all of my projects are very diverse and different, and I feel like I like them all for different things. Most notably my metadata study is my favorite right now because it's done. So hopefully that answers your question. I find joy in diverse things. Yes, Ed?
Yeah, I was kind of, when you have leaves that aren't hairy, is the infection, can you see it from either side of the leaf fairly equally well or not?
So that, yeah, so Ed is asking about the phenotypic expression of the disease on the leaf disc and if we can see it on both sides. So that depends on the pathogen. So my pathogen, downy mildew, only sporulates from the bottom of the leaf. So on the top of the leaf in the field, you'll see some like oil spotting or lesions, but they won't sporulate from the top. So on the blackbird, we always phenotype from the bottom and we always count severity as the little fluffy sporangia that pop out. In terms of powdery mildew only like sporulates on the top of the leaf. So when we do those in the blackbird scheme, we put them right side up to observe disease from the top. So again, dependent on the disease that you're looking at.
Because I was wondering if there's some way you could play with light, its transmission, and flipping the leaf, training a model, so you train on one side and the reverse side, and then with transmission, if you could somehow pick it up, because, you know, I agree, the refraction off those leaves may be, or off the trichomes may be tricky.
Right. Ed is offering that instead of looking through the leaf hairs, if I flip the disc over and observe some type of phenotype from the top. I mean, we haven't tried, especially for the blackbird scheme, they're sitting on agar. So if you were to put downy mildew upside down, I don't even know if that would like kill deomycete because you're cutting it off from the outside, you're not letting it breathe. In the field, sure. I could see like you see symptoms on both sides of the leaves, but I'm not sure on the blackbird system whether that would be feasible. Yes.
You see the January paper on it was out of Julius Kuhn where they did a ResNet for like RGB imaging of leaf hairs, and if so, what are your thoughts on
Is that the one out of JKI?
Yeah.
I have different. I have read that one. I do like that one. I'm sure that's great. We have a different. We have a different CNN that was made by someone else that, like I have and know works. So I'm gonna train against that. But I'm sure that one's great and fine. I love Oliver Trapp and the people that work at JKI. So, again, sure that's great.
This has been a production of Cornell University, on the web at cornell.edu.